The overall goal of this surgical intervention is to observe the effect of the vagus nerve stimulation in inflammatory conditions, to analyze and understand the different mechanisms and cells involved in the so-called cholinergic anti-inflammatory pathway.
This method was developed in the Kevin Tracy lab in the Feinstein Institute. The method can help answer key questions in the neuroimmunology field, as recent advances establish a close link between the nervous and the immune systems. The main advantage with this technique is that it can be easily set up in a laboratory. But important rules have to be followed regarding animal care and the surgery protocol.
The wandering course of the vagus nerve throughout the whole body and its ability to downregulate inflammation in various animal models implies that its stimulation could be useful in the treatment of many inflammatory diseases.
Usually, individuals new to this technique will struggle to identify and isolate the vagus nerve from the carotid artery under microscopic observation. However, though it is tricky at the beginning, it becomes easier with time and practice.
Begin by turning on the computer and the data acquisition system linked to the stimulating electrode. Launch the data acquisition and analysis software. Then, thaw an aliquot of 5 mg per milliliter lipopolysaccharide, or LPS, and dilute it to 0.5 mg per milliliter with saline.